Good afternoon, everyone. I hope you're enjoying the conference and stay this late. I want to really thank the organizers for inviting me and giving me the opportunity to teach you a little bit about our research. So my talk is called Structural Basis for Ryanodine Receptor 2 Leak in Heart Failure and Arrhythmogenic Disorders. So I will be talking about one channel, which is ryanodine receptor, the cardiac isoform, two diseases, mainly heart failure and inherited arrhythmias, and then one fits that we are developing a drug to target both diseases. So my name is Miotto. I'm an assistant professor at the Department of Physiology and Cellular Biophysics at Columbia University in the city of New York. So let's start. So as you've been talking these couple of days, we have heart failure, which is an acquired disease. Most of these patients have, as you know, problem pumping, but the failure of the pumping is not the cause of death. Usually, patients die because they have a sudden arrhythmia or tachycardia. And we have other types of patients where they have inherited arrhythmias. And we in particular study CPVT, which is catecholaminergic polymorphic ventricular tachycardia. So in this case, healthy kids, or what appears to be healthy kids, collapse during sport or during intense emotional stress. The heart is structurally normal, but yet the arrhythmia that they suffer is lethal. So they have the same terminal event, right? This is a trigger arrhythmia driven by calcium leaking out of the sarcoplasmic reticulum during diastole, right? So we have during diastole, where the heart should be silent, we have leak of calcium that triggers contraction. So we have different clinical states, right? Heart failure and inherited arrhythmias, clearly different genetics, and one molecular endpoint, which is the calcium leak from the sarcoplasmic reticulum. And that's exactly where the structural biology, and what we do at the atomic level can help. So ryanodine receptor 2, or RWARE2, right, as we're going to call it, is the calcium switch behind every beat. So to remind you a little bit of the physiology or physiognomy of cardiomyocytes, we have the T-tubule where the L-type calcium channels are, right? The first step on the contraction on every beat is the action potential that comes through the plasma membrane and activates the L-type calcium channels. Calcium enters through the L-type calcium channels and activates RWARE2 in a phenomenon called calcium-induced calcium release. So calcium is the activator that opens RWARE2 to lead and release more calcium from the inside of the sarcoplasmic reticulum. And then this calcium, which can go to the cytoplasm and binds to the myofilaments, induces the contraction of the contractility machinery. And we can see here the action potential. We have like the three phases are kind of like delayed, so timing of the calcium wave is really important. And the job of RWARE2 is timing. It needs to be open during systole and needs to stay shut during diastole. What happened in disease is that, this is how it should work. Normally, everything is coordinated. In disease or in arrhythmia, some of the cardiomyocytes start leaking calcium, and the leak without the action potential will still generate a contraction of the myofilaments, but all the contraction of these cardiomyocytes will be independent and will not be coordinated. And then we have arrhythmias, tachycardia, fibrillation, and this is really dangerous for, this could be completely deadly, right? So the question and how we can finally answer it. So both diseases show AWARE2 leak. These have been proven by many biochemical and physiological studies. So we were wondering if this leak that is observed both in heart failure and in hereditary arrhythmias share the same defect at the atomic level. So this is the structure, the atomic structure of AWARE2. This is a very big 2.3 megadalton machine. It's a tetramer and it's way too dynamic for crystallization. So we use cryo-electron microscopy, cryo-EM. We freeze single channels and we average thousands of particles into a 3D map. And with the 3D map, we build atom by atom, we build this atomic model, which we can understand and study. So for you to have an idea of how this looks like, we purify first AWARE2. We can do it from heart tissue or recombinantly from cells, and that allows us to also purify mutants. That's great to study inherited arrhythmias. And we purify a lot of this of AWARE2 and we put it on a small grid, and then we take electron microscopy images. So the image looks like this, and you can see it's completely covered with this small shape that looks like ryanodine receptor, and actually each one of these is one particle. So we select and extract all of these particles, we average them, and because they have all the orientations, we can create a three-dimensional map that looks basically like this. And on this map, we're going to build the atomic structure. So AWARE2 works, has mainly two multi-domains. So we have the cytoplasmic shell or the regulatory shell. There's this big part of the ryanodine receptor, and it has an upward downward movement. It's like breathing. And then we have the transmembrane domain, where we have the pore, which opens and closes so it can release calcium. And this movement, this breathing movement and the opening and closing is coordinated. So when the wings or when the cytoplasmic shell is up, the pore is closed, and when it's going down, the pore opens. So we can think of as a wing corkscrew, right? The wings are up and then the bottle remains closed, and when you pull the wings down, you can open the bottle and then calcium leaks. But this process needs to be highly regulated because you want to open the bottle only on the weekend, not every day, right? So we studied, like I mentioned, we purify different samples, AWARE2 from inherited arrhythmias, different mutations that have been proven to be very aggressive in patients, and also we did purification of the wild-type AWARE2 and introduced the modifications that are found in heart failure. And to our surprise, all of our disease proteins that we resolved through cryo-EM were in third state, not open, not closed, somewhere in between, that we call the prime state. So the prime state is special because the transmembrane domain, here we have overlapped the wild type in gray and the mutant in purple. We can see that here the two structures completely overlap, so the closed and prime state look exactly the same on the transmembrane domain, but the cytoplasmic shell on the mutant channels are moving downward, right? And that's similar to what happens on the cytoplasmic shell of the open state. So the pore is still shut, but the regulatory shell has already shifted halfway down. So we can think that the wings are almost completely down, but we can see that the bottle remains closed. So even if it was genetic or acquired, the channel is called poised to open, so the prime state. We then studied different triggers, right? And they all land on the same prime state. We tested genetic mutations from CPVT, R2474S, R420W, R420Q. They all land in the prime state. We did heart failure remodeling, where the protein, the channel is oxidized, and one of the subunits called calcium 2, which is a stabilizing subunit, is detached and it's lost. And then we have hyperphosphorylation, where adrenaline or catecholamines induce the activation of the kinase PKA, especially on residue 28OA. So we mutate this residue to 20OA-D, the aspartate with negative charge, to simulate hyperphosphorylation. And we saw that we resolved the cryogenic structure of all these different conditions. They all lead to the prime state, where the shell is shifted halfway toward open. So this prime state, as we were talking, causes diastolic leak and arrhythmias. So we have three completely unrelated independent insults, and they share one conformation, and that's a unifying mechanism. So why the prime state means leaky, and why stress is triggered? We have actually energetics, right? It's an energy picture. We have that the closed and open state are in two valleys, right? Local minimum, but they have like different in energy, right? And to go from one state to the other, they need to climb a hill, right? They need to go through an energy barrier. So what the prime state is doing is basically chopping the top of the hill, making the climbing to the other side easier. So a much smaller push now tips it open during diastole and leads to a spontaneous leak. Actually, what it's doing is shifting the difference of energy between the two states, so the actual energy barrier is smaller. So here in wild type, we need to go from here to here, from the closed to the open state, so the energy barrier is this big. But on the prime state, the energy barrier is much smaller, so going from the closed to the open state is much easier, and we have more leaky channels. And the push that moves one to the other is actually adrenaline. Exercise and emotional stress raise catecholamines exactly when CPVT kills, and exactly the hyperadrenergic state of heart failure. So the structure explains the trigger that the patients already report. So because they all share the prime state, we said, well, maybe a drug is able to reset the channel and fits the cytoplasmic shell and move it upward toward the closed state. So we developed Rycal drugs. The one that we're studying right now is called ARM210, which binds on the outer part of the cytoplasmic shell, and it's able to push the shell back towards the closed state. It pushes it up. So we can see this here in cyan on the cryo structure that we resolve. So we obtain less diastolic calcium leak, better contractility, and fewer arrhythmias. So we quantify the movement of the cytoplasmic shell, right, on each of our different structures. Here we're showing this in purple. So we tested the three CPVT variants that we have. They all have a prime state, but each of them is different, right? the level of movement of the cytoplasmic shell is different, and also the phosphorylation. And when we add arm-to-10, here in blue, we see that they all decrease the same amount. So this is saying that the effect of arm-to-10 is constant. The energy that the drug is giving to the cytoplasmic shell to move upward is the same, independent of the insult that generated the prime state in the first place. So we have one drug strategy that works regardless of which trigger caused the leak. But then we wondered, well, patients don't suffer arrhythmias all the time. There must be a natural inhibitor that is modulating this cytoplasmic shell. And we found that inhibitor is calmodulin. So calmodulin is able to bind, here we see calmodulin in orange, is able to bind to this side of the cytoplasmic shell. And when it's bound, we can see that the prime state in purple gets reversed towards the closed state in blue. So it has the same effect as the Rycal drugs. But our hypothesis is that calmodulin during stress conditions like heavy oxidation, nitrosylation, glycosylation, or imbalance of protein synthesis, which can diminish the concentration of calmodulin or can sequester calmodulin to other parts of the cell. So in these cases, calmodulin will have less affinity and more of our RR2 channels will be found in the prime state, being able to leak calcium during diastole and lead to arrhythmias. So the whole story in one picture. We have healthy RYR2, where casdatifan is bound. We have that it gets activated by calcium and inhibited by calmodulin. But on disease, we have the prime state, where calmodulin has gone off and stress conditions themselves can open the channel and lead to leaky RYR2. And this gets inhibited by Rycals. So the whole story is actually thermodynamics, right? We have this continuous motion of the cytoplasmic shell, and we can see that some insults like PK phosphorylation or mutations can shift the difference between the values, but we have also Rycals or casdatifan or calmodulin here that can make them farther apart, so it takes more energy to go from the closed state to the open state, and the leak gets reduced. So for the clinic, we are proposing a unifying mechanism where the arrhythmias from CPVT, heart failure, and probably atrial fibrillation share one structural defect, which is RYR2 in the prime state. Then we have a druggable target stabilizing the closed state. Rycals reverses leak as its source, and it's a channel conformation therapy, which now is under clinical development. And we have a new way to read the disease, not by genotype or acquired, but on channel conformation. So to conclude, the RYR2 prime state is the common structural basis of calcium leak in both hereditary arrhythmia syndromes and heart failure. Diverse triggers, mutations, phosphorylation, casdatifan loss converge on it, and Rycal drugs reverse it toward the closed state. And cryo-EM turns what looks into a clinical spectrum into one mechanism that you can target, the prime state. I want to thank my colleagues at the Marks Lab at Columbia University, my colleagues also at the Department of Physiology and Cellular Biophysics, the cryo-EM facility, and of course the NIH funding that allows all this research. I want to thank you. I'm open to questions, and if you have inquiries, also you can email me later to mm5642@cumc.columbia.edu, and I'll be happy to chat with you and answer more questions.